Okay, so today is our 22nd lecture. So we are going to talk about microscopic data analysis in the sense that so far we have seen omics data, but we haven't seen kind of imaging data yet. So in this lecture, we are going to go over what kind of imaging data we see in biology and how do we extract information from image and what are computational tools or techniques that will help us kind of make quantitative assessment of image in biology. So today's agenda, we're going to talk about the concept of microscopy, which I think you are more familiar than me as a wet lab person. We're going to talk about how we can extract biological features or biological knowledge from microscopic image. And we're going to go over some image processing concept that whenever you use image analysis tools or whenever you read papers about imaging software, you're going to see these concepts. So hopefully today's content will be kind of beneficial for your work with microscopy as well. Okay, so we know that image can tell us a lot of things. Like when we look at spatial transcriptomics, we see it's not only the expression level of the protein, but also the structure of the tissue. Like we can interpret both the details at the molecular level and the details on the structural level. And when you take kind of imaging like this, like you can see the kind of the shape, the size of the nucleus kind of relative to the cytoplasm. You can mark specific proteins. Sometimes you look at kind of the morphology of the cells without any labels, and you can tell a lot about the disease, like or the condition of the cells by looking at the morphology. So from the perspective of computational biology, we think of microscopy as an inexpensive molecular assay because you can get image with much less budget than omics data. So if you can make the same prediction or you can make the same conclusion using image, it's going to be much cheaper than doing sequencing. And things that are captured through the image are the morphology or the shape or the structure of the cells and the tissue, right? And also the environment of the tissue, like which cells are near each other. Like we can see this type of cell organization as well that we couldn't see with single cells or any other omics data. So when we talk about quantitative analysis of cells, or cells in the image, something that you can imagine is, okay, we have the image and it contains multiple cells, multiple nucleus. How can we segment or how can we isolate the individual cells? So that for each of them, you can quantify the shape, the size, or maybe you can quantify the fluorescence signal. So you can tell whether each cell how many cells express that protein that you state. You have to be able to tell the cells apart in the microscopy image. When we think about spatial transcriptomics, we have two types of information. First is the image. You see the change in tissue structure, like the change in cell type from the image. But you also have the transcriptome data that tells you about the change in gene expression program. So in spatial transcriptomics, you can analyze it in terms of purely omics data, but you can also combine it with the imaging data to tell that we have transcriptomic similarity among cells in this region, like they are all yellow, meaning that they have the same transcriptome profile. But we also want to compare this structure of the yellow cells with similar profiles to their position on the tissue. Do they form the same kind of tissue texture, or do they change, or is there any other type of cells that infiltrate into that region? Like we want to compare both the omics data and the visual data. And currently we have pipeline that can combine section image from your tissues, combine it with spatial transcriptomics or fluorescence barcoding, and you can reconstruct the slice of your tissues together with the position of the cell that express certain genes or protein markers. And nowadays we have tools that can make a 3D projection of your sample. So in terms of morphology, we Even if we don't stain any protein or genes, sometimes just the shape or the size of the cells can be informative, especially for cancer. People who want to detect cancer cells, like in the patient or maybe in the sample, sometimes you don't even need to stain any protein. You can see the size of the nucleus is enlarged. The cell is of irregular shape. These can be a signature of cancer that you can do without any molecular techniques at all. So from the perspective of computational biology, some people want to do image-based classification. By looking at the image, can we develop an algorithm or a model that can classify that this must be a red blood cell, this must be other cell type, for example. And if you can do this, as I mentioned, this is going to be a very cheap for you to deploy in hospital or in the field. You don't need to do omics analysis. So taking this a step further, we not only want to classify the cell type, but we also wonder if we take an unlabeled image, the black and white image, can we train a model that predict the staining? Can we predict if certain protein will be expressed somewhere in this cell? If we can do this, then you don't even need to do the fluorescent labeling. You can just take the brightfield image, unlabeled, and you make the algorithms that can predict if this were to be stained with certain groups of protein, how would it look like? And then you get a cheap molecular assay that you can use to classify the cell type or use to predict the disease. Similarly, some people try to create a model or tools that can help you deal with low quality image. If you take the image and it's blurry, it's out of focus, because it still contains the information of the cell, we don't want to throw it away. Sometimes there are approaches that we can use to predict what would be the original sharp image that gives rise to this noisy, blurry pictures. So these are things that we can train computer to do nowadays. But for this class, we are going to start from the basic. We're going to look at what can we do with image as a human, and then we'll go to how AI or modern software deals with image. So for the first section, we're going to talk about how we can extract biological details, biological features from image. The first thing that we want to be able to do is, usually if we deal with the pictures of the cells, we want to identify the nucleus. Because if you know the nucleus, you know where the cells are, and then you can do other analysis. First, you need to know where the cells are. So the idea is we have an image that contains maybe contrast, just phase contrast image, or maybe standard image. The first thing we want to do is to be able to circle or draw the region for the nucleus or for the individual cells, so that we can analyze other things. So depending on how you take the image, different imaging techniques give you different type of pictures. But for each, there are algorithms that we can do to identify individual position of the nucleus. For example, if we are using a bright field or phase contrast image, we might look at the texture, how the dark area of the image. This might correspond to the nucleus. But if we are looking at maybe some fluorescence staining, in this case, most of the area will be dark, and the bright area will designate the position of the nucleus. So that's usually the first step. We want to identify where the cells are. But once we know where the nucleus is, where the nuclei are, next we want to know the boundary of the cell, so that we know how large it is to a certain extent. And this also depends on how you take the image. For phase contrast, you sometimes see the boundary of the cells being either very dark or very bright because of the contrast. So you can use some image processing algorithm that can detect the boundary. So in later slides, we will talk more about the details of this algorithm, and we are going from the high level into the low level. But if you're taking a fluorescence image, if you stain cytoplasmic protein, so basically this green, dark, bright green area, these will designate the expression of protein that are in the cytoplasm. So the radius that you see in this image, these are the boundary of the cells, because the protein can go anywhere in the cytoplasm. So you're going to see the entire range of the cell. So you can use kind of these features or these characteristics to guide the analysis of the image. But sometimes, and this might happen to you in the future, in some imaging techniques, we might stain some of the protein, but it's not good enough to tell you the entire region of the cells. In this case, you only see like the core of the cells where the proteins are highly expressed. But you can, if you look at it carefully, you can see that actually the cells kind of span the area beyond that. So sometimes we need to do some approximation, and this will happen a lot in image processing algorithms. So for example, in this case, like we can tell the center of the cell very easily because the signals are bright. But then if we want to identify the actual radius of the cell, we have to make some assumption because we don't have enough information. So in this case, algorithm like this, the one shown in this panel, basically assume that the cells are clustered together. So we are seeing like a clump of cells, and each cells are roughly the same size. So basically from each kind of center of the cells that we identify using the bright signal, we're going to grow the area, let the area expand until it touches the other. So you can imagine that if you start with this image and you expand all the cells kind of outward until they touch each other, you're going to get something like this, like a beehive, that you get all the position of the individual cells kind of located. So sometimes in the algorithm, we need to make this kind of assumption to get an approximation of where the cells and the nucleus are. Okay. So once we can locate like where the nucleus are, where the cells are, like next would be to try to understand if the cells are the same type, do they express the same protein? So in this case, you are going to analyze fluorescence data usually. You stain each protein with a different fluorescent color, and when you activate the fluorescence for that protein, you're going to see one image for each protein. So in this case, usually blue is for the nucleus, and the yellow, green, red, like these are for different proteins. So once we are able to identify like the location of the nucleus, like maybe using the blue color, identify the boundary of the cells using the propagation, like we assume that cells are next to each other. Then we can ask like for each cell region in the border, what's the expression level for the protein? We might look at the total intensity for the protein, for the green protein that we stain with the green color. For the yellow protein, we can ask how intense are the signal inside the cell. We can quantify the protein expression. But for some protein, they aggregate it together. So if you compare the patterns from the top example to the bottom example, the top example, these are proteins that are diffused across the cytoplasm or across some organelle. So you see a smooth shade of colors throughout. But some protein aggregate, and when you stain this protein, they will appear as dots in your image. So some algorithms will allow you to either quantify the average or total intensity inside the cells, or you might want to count the number of dots, the number of spots for the protein. These are for like different type of protein. But sometimes, like if you recall back to the session about system biology, we mentioned that in the body of the animal, different protein must be expressed. Some proteins are expressed at the head region, some proteins are expressed in the tail region. So in image analysis algorithms, they also have to account for the possibility that maybe in your cells, the protein are not expressed kind of all throughout the cytoplasm, but maybe they are expressed toward a certain direction, maybe in contact with a different cell type, or maybe in the tissue structure. So in some of these algorithms, they're going to allow you to analyze the polarity of protein expression. For example, let's say this is the nucleus of the cell, and this is the boundary of the cell. So some proteins are expressed as a gradient. They are highly expressed near the nucleus, but not highly expressed around the cell membrane. Some proteins are localized near the nucleus. On the other hand, some proteins might be highly expressed in one direction of the cell, because this side of the cell might be in contact with different cell types. Like there are some cell-cell communication, or in the structure of the tissues. Like this side might be one tissue type, and the other side might be a different tissue. So in this case, you can detect whether the expression of the protein is it homogeneous across the whole cytoplasm, or is it there is a radial pattern from the center to the outward, or is it like polarized? One side has more expression than the other. These are patterns that algorithms can detect in the image based on these fluorescence signals. The other thing that we can analyze about the cells is the texture. In this case, texture you can think of as a roughness of the color. So if you compare these untreated cells, if you focus on the protein that are stained with the yellow fluorescent, yellow dye, you can see that in the untreated cell, you can see the fiber of the protein very clearly. But in the treated cells, you don't see as much fiber structure as before. So in this case, in terms of calculation, if we focus only on the yellow color, highlight only the yellow color, we can tell that in the untreated cells, like the image looks very rough, like you have bright and dark, like change in color everywhere because of the fiber structure. But in the treated cells, because the fiber structure collapse, you see less change in color throughout. So this difference, like the change in color, not the intensity, but the change in color. These are in the computational terminology, we call this texture of the image. So you can think of as roughness, like how the color change, or the smoothness. In the untreated cells, like the color is not smooth because you see bright and dark pixels all over the place. But in the treated cells, you see bright pixels kind of clustered together and dark pixels clustered together. So texture, in terms of computation, is described by the histogram, like the frequency. But instead of the frequency of intensity, it's going to be a frequency of difference in intensity. Instead of the actual intensity, it will be a difference or a change in intensity. So to give you some example of the texture features. So for example, if we want to compare adjacent pixels, we want to compare pixels that are next to each other along the top and bottom direction. So in this case, we see there are 3 places in the image that have 1 and 2 next to each other. So in the histogram, the first row and the second column, we're going to put 3 in here, because 3 is the number of pixel pair that have 1 and 2 next to each other. And 1 and 3, this is more change, more change from 1 to 3. It's more change than from 1 to 2. There are 2 places in the image. So in this case, the first row and the third column of our texture data will be 2 that designate these numbers of pixel pair. So what you can see here, the diagonal of the texture means that the bottom panel, the bottom pixel and the top pixel are the same. 1 and 1 together, 2 and 2 together, 3 and 3 together. So the numbers along the main diagonal means that there is no change in intensity. Meaning that if you have a lot of pixel pair in the diagonal, if the numbers are high, that means your image is smooth. There is no change in the pixel intensity. But if you have a lot of off diagonal entry, like 3, 2, 1, 2, 2, 2 here, means that everywhere in your image, if you take the two adjacent pixels, their intensity are different. In this case. And if you are going further away from the diagonal, the more change in pixel intensity you are observing. So this kind of a histogram can tell you how smooth or how rough the pixel intensities in the image are. Look at another example. So this histogram is about how long can you find pixels that are next to each other and they are of the same number. So in this case, if we want to find how many pixels in this image that have the intensity value of 1 and they are next to another pixel with the same intensity of 1. Like there is only one here, 1 and 1. And there is one place where we have pixel with the same intensity, 1, 1, 1 together. So in this case, this metric tells us that if we have more and more off diagonal numbers, like in the histogram, this means that you have many pixels with the same intensity in your image next to each other. So this is opposite to the top texture. Like the top texture, numbers in the diagonal means that you have the same intensity in your image. But in the bottom texture, histogram, the numbers in the number on the first column means that you have singletons in your intensity, like 1 next to different intensity, 1 next to different intensity. So in this case, the number in the first column, if they are high, it means that your image is very rough. Every pixel is next to a pixel with different color, different intensity. But if you have high numbers in the large letter and the letter columns, this means that you have pixels with the same intensity next to each other, meaning that you have the smooth color in the image. So many people use this type of calculation to describe if we are seeing a rough image or being a smooth image. And this can be used to describe the state of the cells or the state of the protein expression. So basically, like these are how mathematicians or engineers try to quantify the characteristics in the image that usually as biologists, like you describe with words. Like you say that this protein forms a fiber, and you have a broader fiber structure in the untreated condition, and you have less fiber structure in the treated condition. But if you want to do computational analysis, you have to be able to transform that description into numbers. And this kind of histogram calculation is one way to turn the description of fiber structure and smooth or rough change in the color into something that can be described as numbers, so that we can develop algorithms, develop models for. So if you use like software that can put in like a microscopic image and then produce that kind of histogram or description, numerical description of the image, they're going to describe many things for you. For example, they can describe for each cells and nucleus, like they can describe the shape, the geometry of the geometric shape of the cells and the nucleus, the area, for example. And if you have a staining of proteins or RNA, they can describe the intensity. They can describe the average intensity. They can describe the radial, like the radial structure of the intensity, like this. They can also describe the polarity of the intensity using this idea. So you're going to be able to produce many numerical descriptions of a single cells in the image. And the texture, the one that we just mentioned, this number can describe how distributed the protein inside the cell. And you can also even calculate the correlation of these features, of these shape, size, intensity, and texture between adjacent cells, so that you can tell whether cells that are next to each other, do they have the same morphology, or do cells next to each other have a different morphology. That can be an indication of the tissue organization. So these numbers are the ones that you can use to characterize the image. But there are a lot. If you I didn't put in the numbers here, but in many software, if you run, you will get more than 800 to maybe 1,000 numbers, different numbers that fall into some of these categories that describe the cells that you have in the image. So the next thing that you need to be able to do if you want to analyze this data is how to screen, how to select only the useful features out of 1,000, which one is useful for describing the cell's condition or describing the disease state. So this idea, I want to highlight that is in omics, we also call a shotgun approach. Like when we think about shotgun sequencing, like shotgun metagenomics, the term shotgun means that you are shooting with a whole bunch of bullets. Instead of targeting one gene, we're going to sequence all the genes. Instead of amplifying or culture just one bacteria, we're going to sequence all the bacteria. So the shotgun approach, you are already familiar with in terms of the omics technology. But in the image analysis, this idea of producing thousands of descriptions from different perspectives of the cells can also be thought of as a shotgun technique. Like we don't know if the area changed because of the disease. We don't know if the protein expression changed because of the disease. So we just compute everything. And we need to figure out kind of which image features are the ones that are useful. If you think back to the transcriptomic lectures, we also had to do this before when we perform differential expression. If we are essentially trying to weed out all the unrelated genes and then only focus on genes that are differentially expressed across the condition. So we have to do the same thing here. We can do univariate analysis, like differential expression, where we compare the shape, the volume, the expression level of the protein across cells from different conditions. You have image from untreated condition, you have image from treated condition. You have the shape, area, intensity, texture, measurement from both conditions. Now you can compare if those numbers differ from across the condition. But in this case, unlike the model that we learned so far, we have a Poisson model for certain type of data. We have negative binomial distribution model for read count. But for these numbers, the size of the cell, the texture of the cells, we have no idea what distribution describes them. So most of the time, we're going to rely on non-parametric tests, like Mann-Whitney U-test or Wilcoxon test or Kruskal-Wallis, because this doesn't assume that the numbers are distributed according to any known distribution, and that's the situation that we have. So this is one possibility. You can do a lot of univariate analysis, and you will get to see which one are statistically significant difference across image from different conditions or disease. The second technique that I want to mention here, and you will see this in some papers that use data-driven or machine learning techniques. It's a not univariate, but it's a multivariate feature selection. So in this case, there are usually two main strategies. You can start with a model that doesn't have any features. Basically, you are predicting just the mean of the two samples. And then you're going to expand your model by adding one parameter. If you think of this as a gene expression model, you start off with a null model with no genes. You are just guessing. But once you know the expression level of one gene, if it's a gene that are different, significantly different between sample, between treatment, if you know the expression level of this gene, you can predict if the cells are from one condition or the other. But if you pick a wrong gene, you pick housekeeping gene, that gene will not help you at all when you want to classify or to predict. So the idea of forward selection is you're going to try, if you have 1,000 genes or if you have 1,000 image-based numbers, you're going to try picking one column at a time. So it's a brute force approach. You're going to pick one column at a time. And whichever column that allow you to distinguish cells from different conditions, you're going to keep that. Let's say the the genes or the features in blue, like this is the columns in the data that allow you to distinguish between cells from different conditions. So you will keep this. This one is the best marker you have. Then you will try to build a multivariate model by looking among the remaining features, the remaining columns, which one that you can pick, and then it works the best together with the blue genes or the blue features. So basically you try to grow your model one column at a time and check if adding this improves your ability to distinguish cells from different conditions. If it does, you keep it. And then you look among these remaining features or genes for the next one to add. But if none of these help you, like you stop. And then you can say that, okay, in my data set, I need only these two genes, or I need only these two image features to distinguish my conditions. That's one approach. Like you start with nothing, and you try to add the best column in your data. The other idea is you start with the full data. You use all the columns to build your model. So basically, your model is going to be very large. You will have a lot of coefficients for all these numbers, like thousand numbers. Then, what we are going to see, maybe not now, but in the future lectures about machine learning, when you build a multivariate model, whichever model you use, there will be a technique that will allow you to tell among these columns, among these inputs, which one is the worst, which one contributes the least. We can identify. For example, maybe the yellow gene or the yellow features from the image is the one that doesn't help you distinguish cells from different conditions. Using the backward idea, we will remove the yellow gene, the yellow column from the data. So we now have one less input. And we'll keep doing this. We will keep removing the least contributing features or the least contributing columns until toward the end, like you can imagine that if you remove columns from your data, like one at a time, to a certain point, there will be a certain point in the process where every column that remains are important. So this process, there will be a certain point that everything that you have now, that you have left, are all important. So if you remove some of them, you're going to lose the ability to distinguish the set. So at a certain point, you will stop removing the columns, because once you remove the next column, your ability to distinguish cells decrease. So this process has a well-defined kind of steps, what to do, and they have a well-defined stop criteria, like when to stop doing this. So this idea, we will see again in the data-driven lecture, can help you go from 1,000 columns, maybe down to just 5 or 10 columns. So basically, you don't have to worry. Even though we are producing, some software can produce more than 1,000 columns for you, there is a way for you to remove the uninteresting columns, the unimportant column, and retain the one that you need. There will be a feature for that. Okay, so for the next section, we're going to try to understand, like when we mentioned that we can detect the individual nucleus, that we can identify the individual cells, how exactly can we do? So this will fall into a field called image processing. So it's similar to how you edit your picture. Like if you use photo editing software, like you can highlight the area of your face, like you can change the brightness, you can change the contrast of your image. The same thing applies to biological image. But instead of detecting your face, we're going to be detecting the nucleus or the cell. Instead of changing the contrast, we're going to try to identify the expression of the protein, for example. So let's see. So let's think about the nuclei detection process. So let's say we have this kind of H&E stained image. So we know that the purple, the dark purple area, these correspond to the nucleus. So maybe you can use the color, and try to distinguish pink from purple. let's say you put a threshold on how purple a pixel is. You can identify areas that are purple. But there can be problems, like some of these nucleus, because they are next to each other, like when you identify them as using the threshold on the purple, like they are touching each other. So instead of seeing the individual nucleus, like you are seeing a connected connection of multiple nucleus together as a single object. So next, what you need to be able to do is to try to cut these touching nucleus, these nucleus that touch, like, into individual ones, so that you can specify the position of each cell. So this process, if we were to summarize, like, we can start by thresholding on the signal that tells us where the nucleus is. Like, it could be the color from the H&E stain. It can be the Darby channel in the fluorescence. But we still have nuclei that touch each other. So these are called clumped, object clumped together, or object occluding each other. So sometimes they are on top of each other. So we need to be able to divide, like, to split this object. And then, like, you can see that there's still some noises. Like, we have very small purple-ish region that these are obviously too small to be nucleus. So we can do filtering by size to remove these. Okay. Let's look at these three sets in more detail. What happened to the image? Okay. So let me show you some example. When we think about the image, to keep it simple, we're going to use a grayscale image in this example. But basically, you can think of color image as multiple versions of the grayscale image, one describing different colors. So basically, when we look at a grayscale image, one thing that you can do is to plot the histogram of the intensity. So in this case, like in this image, most of the background have the same color, and you only have a small bright area and a small dark area. So when you plot the histogram, it's going to look like this. You have a very high frequency of pixels with the same color, and you have very few pixels with dark or light color. This is a bad histogram, because when you see all the pixels, like, being the same color, this also means that you wouldn't see much of a change, because everything looks the same. So there are ways that we can change the histogram, either by multiplying the intensity with some constant, by putting a threshold on the intensity at some constant. We can adjust the histogram to make it broader, to make it higher, to make it lower, to make it more uniform. We can make this adjustment. So these are something that usual photo processing also does as well. You can change the contrast of the image to be able to better see individual objects inside. You start from this original that have low contrast, but by adjusting the intensity range, how it's distributed, we are able to see better that there are multiple objects in this area. We can also use this technique to help highlight maybe the fluorescent image are correct, kind of took with low exposure. Like, you don't see anything in this region. But if you adjust the histogram of the brightness, you will see the fiber, the structure of the dark area. So this kind of a histogram normalization or histogram modification can help us see better details in the dark area or the bright area of the image. Okay. So next, like we say that we want to be able to detect the nucleus, like, by putting on some threshold. The area that are purple, we think they are nucleus. The area that have a high Darby signal, we think they are nucleus. So after we make the adjustment to the histogram, like, to see the image better, then we can try to select based on the histogram where to put the threshold. If the signal is above this threshold, we think that they are nucleus. If the signal is below the threshold, we think that they are just background. or noise. So one of the assumptions that people use to make this kind of cutoff is we assume that, let's say if we focus on the purple and the pink color, right? In the H&E space, that there's only two primary colors. So if we look at how purple, like, the pixel is, we can imagine some kind of distribution like this, where the nucleus, like, will look purple, like very purple, very high intensity. But the other area of the image will not look purple at all. So you should have many pixels with low intensity in terms of how purple it is. So we can use this assumption to say that the region between the two peaks, like one peak is for the nucleus, one peak is for the rest of the pixel. We can imagine that this cutoff in the middle is probably a good cutoff to identify our nucleus. So these are just some examples of how this kind of thresholding can help you clean up the original image that may have a variable in signals, but we want to identify individual objects from. These are just different types of cells or structure inside the image. So that's the first part, that we can use this kind of threshold to at least flag where the nucleus should be. So next, we have to deal with this problem of touching nucleus. How do we cut them apart? There are two ideas that people use to isolate two objects that are touching each other. In this case, we use the assumption that cells or nucleus are usually can be described as an oval in general. We think of it as an oval shape. So when you have two ovals that touch each other, so for example, in this conformation, there will be two things that will help you distinguish that there are two objects here. First, like, there will be some indentation, the contour. The contour of the oval is smooth and has one kind of convex pattern. But if you have a change in convexity, you have an indentation, that's an indication that you have more than one object. Or if you are staining, like with Darby maybe, your nucleus, like, or any other object, like when you stain, sometimes there are more, the signal is brighter in the middle and less bright towards the periphery of the object. So if you have two objects that touch each other, you're going to see two centers of intensity, and then at the boundary here, it will be low signal. So people can use change in intensity along this object to tell that this drop in intensity is probably the boundary between the two objects. So when you run the algorithm, they will ask you either to use shape or use intensity to distinguish individual objects that touch. Okay. So once you are able to isolate all the individual nuclei, the next step would be how to define the cell. So in this case, there is a cool algorithm that I want to introduce. It's called a Watershed algorithm. So for the idea behind the Watershed algorithm is, so let's say you have, let's say you identify the position of the nuclei, like you isolate them into individual nuclei. In this case, we're going to flip the color. Instead of the black background and the bright foreground, we're going to flip the color to make the foreground brighter and the nucleus darker. Because we're going to imagine this kind of geographic, so we're going to turn this image of a cell into a geographic map. The dark area means there is a hole. The bright area means it's high. So in this case, each nucleus that we detect will serve as like let's say a water reservoir, like a lake, for example. So if you fill water into this map, you pour water into this map, it's going to fill the hole first, and individual nuclei, if we color them with different, if we put them with different color, water that goes into nuclei number 1, let's say they are red. Water that go into nuclei number 2, let's say they are blue. When we pour color into it, you can see this kind of pattern. The red color will fill in this part of the geography. The cyan color will fill in this part of the geography, and the blue color will fill this part of the geography. So you can turn this back into cell boundary. So we say that the region that the red water can reach, this is the cell that we will call the red cell. The region that the cyan color can reach, this we will call the cyan cell. So this kind of basically is the idea that we assume that the cells are next to each other and they are touching each other. And the center of each cell are these bottoms of the reservoir, are these nuclei. So when we fill water, we are basically drawing the radius that the water meets each other. So you can think of this as like drawing a midpoint between two nuclei to divide them into the cell. But instead of drawing the midpoint as a direct line, we try to use the signal in the data. Even though these are all bright, right, it's not all the same intensity. Some are slightly gray, right, some are slightly white. So it can be thought of as a different height in the geography. So this kind of watershed algorithm is like you pour water into the system and see how the water segregates into different reservoirs. And that the boundary between reservoir is where you draw the position of the cell. So it's a cool idea that allows you to identify cells with the idea, with like a physical simulation. Okay. But sometimes, using these type of algorithms, you're not going to get a perfect boundary or perfect positioning of the cell, of the nucleus. Sometimes, because the fluorescence signal can be very spotty, no matter how you try to threshold, or no matter how you try to fix them, you still see spotty signal. So the next type of techniques that we want to talk about will be how can we connect these fragmented, these dotted objects. Because by eye, you can tell that this must form a single nucleus, a single cell. But because your signal are so spotty, no matter how you analyze, you see separate small particles. Or sometimes you might have a region in the cells that are not stained very well. It might be a separate organelle, something inside the cell, and you have a dark spot. And you want to fill this, you want to fill the hole in your segmentation. So how do we do this? These are the operations that can solve this problem, are called morphological operations, because they deal with the morphology of the object. And there are 4 keywords that I want to introduce, but hopefully you will see that they are very logical. So they are erosion, dilation, opening, and closing. So let's see what they are, using this image as an example. So this is the original image. It has 2 features that we want to address. First, there are holes in this part. There's areas that are not fully black. And there are also small noises from this region, like tentacles, that we want to remove. These 4 operations, the fact that they are interesting is if you perform erosion, the idea of erosion, as the English term implies, you are removing the pixel from this image, from this object. So from this object, you are removing the black pixel by setting it to white from the outside in. So you can see that the object became thinner. You, instead of having multiple pixels in black, now you have only 1 pixel in black. But the good thing is, because the object is thick on this side, but it's very thin on this side. So this part will be completely gone, because if we remove enough pixels, then they are gone. But this part is still intact, even though they are thinner than before. But if you perform dilation is adding more black pixels to the image outward. So in this case, these two tentacles are now much bigger than before because we add more pixels to it. But on the other hand, the holes on the left side become completely filled because we add pixels inside as well. We add the black pixel inside. So we see that erosion and dilation, very simple operation, thinning the object by 1 pixel at a time or expanding the object by 1 pixel at a time. They solve this same problem. Erosion remove these noises. Dilation fill in the hole. But none of them works by itself. If you do erosion, you remove the noise, but you create a bigger problem with the hole. If you perform dilation, you cover the holes, but you make the noise much larger. So people come up with an idea, a clever idea. Why don't we do erosion and dilation in sequence? So that gave rise to the opening and closing operation. Opening is you perform erosion first and then dilation. Closing is you perform dilation first and then erosion. So what would happen is that if you want to remove the noise on this part, like the tentacle here, you perform erosion first because it will remove that side. But because you want to fill this hole back, you perform dilation to fill in the hole. But if you want to fill the hole first, you perform dilation first and you fill the hole first. But you don't want to make the noise on this part larger, so you perform erosion next. So the noise came back to the same, almost the same size, but you fill the hole. So by doing this operation in sequence, you can solve the problem. So basically, if you want to fill the hole completely on this example, you can do more dilation to the opening, and you will fill this hole without creating these tentacles. And if you want to do it from the closing, if you do more erosion, you get to remove these two tentacles completely without affecting the shape of this object. So these are like cool techniques that people use to make the image look nicer. So for the next part is also image processing, but is a more, let's say, more modern way of image processing, and that's an image processing with kernel. We're going to talk about what kernel is, and nowadays these are also called convolutional operation. So when you heard about convolutional neural networks, these are the operations that happens underneath. Okay, so let's look at what this technique is. So let's look at some simple example. When you want to sharpen your image, if you use Photoshop or maybe there's an app on your phone, you take an image, it's kind of blurry, you want to sharpen it. The way to sharpen it computationally is to multiply the central pixel by a large number. So you want to amplify that signal, and you want to reduce the signal from the adjacent pixel. You want to make it sharper. You want to increase the contrast. So you're basically amplifying the center, and you are reducing the signal from the corner, from the surrounding. So you multiply everywhere in the image with this 3x3 matrix, which with coefficient like 0, -1, 0, -1, 5, -1, 0, -1, 0. So basically, it's going to amplify the central pixels by 5-fold, and then subtract by the sum of the 4 adjacent pixels, and then ignore the corner pixel entirely. If you do this mathematical calculation, which you can discard as a dot product to the image, you're going to produce this sharpened image. In another example, if you want to identify the face, where the face boundary is, you can use this matrix, -1, -1, -1, and then only 1/8 in the middle. This will amplify signal when you have a change in color. So if you analyze the signal around the boundary between the face and the hair, the signal will be high. But if you analyze the signal in the middle of the hair or in the middle of the face, the signal will be low. So by selecting the right coefficients to do the dot product, you can extract interesting information from the image. And these numbers, -1, -1, 8, these combinations are called kernel. So this is one kernel. This is a kernel that tells you the outline of the image. This kernel, -1 and 5, this is a kernel that sharpens the image. So electrical engineering specialized in this type of analysis. They will know which numbers to use to create the image or to identify information in the image that they need. So this operation of taking this kernel, coefficient of numbers, multiply it with the image to extract the new signal. By performing this throughout the image, this is called a convolutional operation. You are applying this matrix everywhere in the image. So you get a new image that has either the outline information or has the sharpened image. One thing that I want to note here, the fact that you are applying the same kernel, like the same coefficients throughout the image, has one implication, which is you don't care where in the image the object is. And this applies for most of your questions, because if you want to identify a cell, it can be in the top corner, it can be in the bottom corner, it can be in the middle. It should still be classified as a cell. So this operation is useful for image because it doesn't care where the object is, and if anywhere in the image, it's going to be detected with the same techniques. So that's a good point. It works. It has this assumption that works well with image. But the problem is, we mentioned that there are two kernels that you now know that works well for sharpening, that works well for identifying the outlines in the image. But how would you know which number is good to identify the nucleus? How would you know which number is good to quantify protein expression? It's impossible for a human to come up with such good coefficient that works for those kind of tasks. So we will learn more about this in the data-driven lecture, but basically we're going to ask the computer to perform the convolution operation, but we're going to ask the computer to come up with these numbers themselves. We're not going to, as a human, we're not going to tell them what the numbers should be in the kernel. We're going to ask the computer that if we give you a lot of image of the cells, can you learn it from the data? So we're going to talk about this more, but the idea is, so let's say you're going to have to do this. Let's say the computer doesn't do this and you have to do it. How would you proceed? You have a lot of image about the cell. With your eye, you know that this is the boundary of the cell. This is the membrane of the cell. Now, your task is to identify the kernel, the right number that can multiply to this part of the image and give you number high score, because you want to tell that this is a membrane. The number that you choose when applied to this region in the image must give you high score. But if you apply it to this dark corner, it should give you low score. If you apply it in the middle, it should give you low score. Let's say you have no idea. You can guess, let's say we want to use a 3x3 kernel. So you have to pick 9 numbers to fill in the kernel. One thing that you can do is you can start with a random number. You can pick 9 random numbers and you try to use it. If it's good, then you are done. But chances are the 9 number that you picked will not work well to identify the cell membrane. In that case, you have a choice in the 9 numbers. Do you want to increase the first number or do you want to decrease the first number? Basically, if you keep doing this, try to increase the first number, try to increase the second, try to decrease the first number, try to decrease the second number. If you adjust this, eventually you're going to arrive at a good set of nine numbers that can help you identify cell membrane. So that's essentially what's going on inside computers. They're going to try changing this number up and down in a smart way until at the end they identify all the kernels that will help them detect that this is a cell membrane, not a cytoplasm, not a vacuum, not a background. And nowadays, just want to show you some example. When people train convolutional neural networks on image data, like image of a car. So what happened inside is that the first kernel, the kernel that I applied directly to this image, is going to detect simple patterns. Like there is a edges or boundary in certain angle, or maybe there is a change in color. The first kernel is not going to be very smart. They're going to identify like simple change in the image. But the idea of convolutional neural networks, if we go back to this diagram, is we are not doing convolutional operation one time. We are not doing it one time. In this example, we only do it one time. But the idea is we are going to do the convolution on top of convolution. So let's say we create a sharpened image. We're going to do convolution on the sharpened image to create the next image. So we're going to do convolution in order, in sequence, multiple times. And in modern models, they might have 100 layers of these. So they're doing convolution hundreds of times. And what happens inside the model is that at the beginning, at the first layers of convolution, they're not very smart. They're just detecting simple change in the image. But the middle layers, the lower layers, people found that when we try to extract what the convolution is doing in the middle of the model or at the bottom of the model, people found that these convolutional neural networks are able to put together these simple lines into geometric objects and eventually into something that resembles parts of a car. It can identify the headlights, it can identify the bumper, and it can identify the wheel. So at the deeper part of convolutional neural network, people found that it's able to reconstruct objects or patterns that are required to classify something as a car. Because if you cannot tell that this image has a wheel, has a door, has a headlight, you shouldn't be able to tell that it is a car because you don't have any knowledge about what constitutes a car. So in this case, we found that for natural photos, the model that are formed like this, algorithms that are developed like this, has the ability to reconstruct human recognizable objects. So when we apply it to cells, they are able to identify characteristics of the cell that are good enough to identify either cell type, maybe growth condition, treat or untreat, or maybe the organelles inside the cell. We're going to kind of go over the idea of these techniques again in later lectures. But for now, I just want you to appreciate the fact that from the image, we know of algorithms that can extract details from the image. But for tasks that are more and more complex, now we have algorithms that allow the computers to learn basically the computer learns the algorithm themselves to extract details from the image, to detect or to classify things that we want to look at. So nowadays, like we have, when you buy a new microscope, sometimes they will come with a software that have a algorithm inside that help you recognize or detect different parts of the image. So for example, in this case, if you want to identify the fiber structure of the protein or the cell inside the image, you can only see with the phase contrast or brightfield data. But if you want to measure how thick are the fiber, how long are the fiber, you need to be able to recognize which part is fiber, which part is not. And nowadays we have imaging software that can apply threshold. The first step, now we know they're going to apply a threshold to identify dark area as potential fiber structure. And it automatically removes the circular object as nucleus. Because the fiber, we imagine that it's elongated, like branching. It shouldn't be oval, it shouldn't be circular. So we can use the circularity of the object to remove the nucleus as well. And we are left with just the fiber part of the protein. And this is the example that I mentioned where in real life, right, we want to understand the cell type or understand the protein expression inside the image. If you want to do that as an experimental biologist, like you have to stain the protein to image it. But nowadays, people are working on models that can look at unstained image, like H&E image, like it doesn't stain for any protein. But because different cell types like have different morphology, and when cells have different undertreatment condition or maybe disease condition, sometimes you can see the change in the physical property of the cell. So based on just H&E image, many software are now able to predict the expression of some genes. And this knowledge will help both biologists and pathologists to understand the cell structure, the disease state, and the effect of the drug, without having to invest resources into staining the protein. So that's everything that I want to talk about today. So see you again next week.